All right. So far I've basically explained to you how does sequence alignment work. We talked about global alignment and motivated how the greedy strategy doesn't work and how dynamic programming strategy essentially works, where essentially the idea is to calculate all the intermediate steps that you require to take in order to go to the best optimization of your problem. And then that was modified to perform local alignments, which essentially is a small tweak in the global alignment algorithm, whereby whenever you are starting a new whenever you are scoring, the minimum score that you can get is zero, right? So zero is the minimum score that you can get at any point of time, which basically allows you to start a new alignment between between any two substrings of the new sequence from anywhere. So I hope this was clear.
Yes, sir.
Yes, sir.
Okay. All right. So now today I'm going to talk about how we can penalize insertions and deletions in a sequence alignment differently. And then I'm going to go into multiple sequence alignment. And before that, I'll also talk slightly about the scoring matrices because I think there was clearly only Ratnesh has tried to read about it and others haven't. So I think uh, I'll spend some time on that. So whenever you are having any kind of sequence modifications in terms of insertions of gaps or deletions in the sequence, they are often of the following four categories. You can have gaps in your genome whereby you have a single gap like this, or you can have a gap in your genome whereby you essentially have a specific like in the middle of the sequence or at the end of the DNA fragment. You can have segmental duplications whereby the same segment, let us say this segment was sort of getting duplicated again and again, or you can have what's called a muted gap where the same particular sequence is now shrunken or it's expanded wherever in whichever direction you look. But in general, um, for in nature, what you see is a series of k indels. Indels essentially means insertions or deletions, often come as a single event rather than k series of single nucleotide events. All right. So essentially, trying to, if you see in all these cases, you are going to essentially see there is much more likelihood that you have single insertion deletion events which are larger in terms of the number of nucleotides than the same number of nucleotides represented as single insertion deletion events randomly spread across, right? So this gives us the idea of actually changing the penalties that you have uh, for gap opening. So whenever you have an insertion and deletion, and the fact that you have already an insertion and deletion, that should now be penalized uh, not as much as starting a new insertion and deletion. So what do I mean by that? So for example, if you have these two cases, in the first case, you have two gaps which are opened, which essentially means that evolutionarily speaking or biologically speaking, there will be two independent events that are happening here, right? So first, there was maybe a breakage of chromosome here, then it was maybe repaired, because of which this particular nucleotide was lost. Then again, there was another breakage of chromosome here, which was repaired, and there was another insertion or deletion happening here. Versus you have a situation like this, whereby there was a single event of chromosome breakage, and while the repairing was happening, instead of one nucleotide, two nucleotides were simultaneously taken off. All right. So now whenever you have these two kinds of events, you see at the end of the day, if the gap, the fact that you have a gap, all both of them are scored equally, then you will get you have possibilities of these two alignments. But biologically speaking, this alignment is a better alignment because it represents biological reality better. So in order to facilitate this, we introduced a concept in scoring called gap opening penalty and gap closing penalty. All right. Which essentially means that whenever you open a gap, a new gap, it will be penalized higher than whenever you are extending an existing gap. So here you opened a gap and you're extending a second gap to the second base. All right. So this should be penalized lower. Therefore, the overall alignment score for this alignment will be higher as opposed to this alignment. All right. So this is what it looks like. So you have this model is called what's called the it's called the affine gap model. This was put forth by the Japanese mathematician Goto, Gotoh. And this is how it said that you essentially would have a penalty uh, for a gap of length k. You will have a penalty sigma, which essentially is called the gap opening penalty, and then you will have a smaller penalty eta, which is called the gap extension penalty. So essentially sigma will be greater than eta. Is this clear to everybody?
Yes, sir.
Yes, sir.
So what does that mean? So essentially means that whenever you're scoring like this, right? This is what you were doing earlier. You were having matches, then you were having insertions or you're having deletions, right? And you were moving in this kind of a direction. But now what you're essentially saying is that whenever you have started to open a gap, whenever you open a gap, the movement or the penalties that you would be having across moving in this direction, you're going to have different kinds of edges possible. When I say different kinds of edges, that essentially means that let us say if you went south on the green edge here once, you can either for the next step, you can either go in this direction, that is you are having a match, or this direction, that is you have having an insertion deletion, or if you are extending this original gap, then you essentially have to have another kind of arrow which shows that you are extending that previous gap, right? So essentially these are the new arrows that you have to introduce. So as a result of which, essentially when you had a matrix like this earlier, where you have moving as insertions, moving as deletions, and moving as matches or mismatches along the diagonal, you would now have for n uh, basically n connected components. Now you are going to add one more layer of arrows, which basically represents these kind of affine gap penalties. So these are the new penalties that you add, and you do that for all the edges in all the directions. So by doing this, you're essentially going from a n squared graph to what's called an n cubed graph, because uh, each node is now connected. There are three ways in which you can go out from, actually four ways in which you can go out from each node. All right, is this clear?
Yes, sir.
Yes, sir.
So are there four ways of going out from each node? I was just testing you. I just randomly said
Three
something.
ways, right?
Are there three ways?
If you're going for insertion,
So
deletion,
ideally five, right? Or six, like depending on how you want to count.
Right. So there is no fixed thing. You just this if you would look at this graph, this now essentially looks like something which is in three dimensions. So if you were to just take it out, so therefore that's why the edges are n cubed here. So now
Yes.
how do we actually represent this? Do we now have to modify the algorithm again? Do we have to now make the algorithm from n squared to n cubed? Is that the case? So long story short, because I've switched the slide once ahead, so you can probably see what we can do is we can actually decompose this complicated graph of your original matrix penalties and the affine gap penalties, right? Which makes a n cubed graph into three n squared graphs. Okay. And this is how it looks like. You have a graph which is basically going to have matches and mismatches. Then you're going to have a graph which is only going to be talking about insertions. And you're going to have a graph which is only going to be talking about deletions. All right. And now if you want to find the best route starting from here to the other end, or for the global alignment, and the same thing For the local element, all you have to do is you have to change the lowest score to 0, right? So what you have to do is at each point, you have to keep track of scores, not only in a single matrix, but in three matrices. How will we be doing it? So let us say we have to go from here to here, right? Let us try to, this was the original path that we wanted to take. We will go from here. So this is a match, let's say. Now, because we see that there is a gap, so now because there is a gap, you start to update these values in the like in one of the insertion or deletion ones. So you go here. Now you basically come back. So going from traveling in the insertion deletion matrices from here back to one of these back to the original mismatch matrix should not have any penalty, right? But the penalties going from mismatch matrix to this one of these matrix gives a penalty of sigma. The penalties of moving in insertion deletion matrices are going to be eta, as you can see here, right? So you move again.
Yes.
Now you go to the other matrix. Again, this is sigma. And moving here, the penalties would be eta, right? And again, there you get a zero penalty, so you come back and then you have a match like this. All right. So simply, if you want to implement this in the in the computer, so you don't have to actually make an n-cubed algorithm. All you have to do is you have to decompose your movement from a single connected graph to three graphs like this. All right. So when you are talking about the lower case, in the case of lower, you basically have to find the maximum of moving of basically moving in the lower matrix, which is lower I comma I comma I minus 1 comma j minus eta, right? And the middle one, right? So middle one is moving here, right? So should you actually uh, open another should you open the gap and go in this direction, right? Or you should move in the case of middle, you already have the values of I comma j that you have already made the move. You're already here, right? So the lower I comma j or the middle. So basically moving from here into this particular node, which is score, right? Which is a math score, or doing the same doing the same movement in the upper matrix. So in the upper matrix, you will basically be figuring out whether your score is the maximum between the upper I comma j. Is this movement the maximum, or is the middle movement the maximum, right? Which is gap opening and opening a new gap is maximum, right? So when you would be implementing it in the case of your algorithm, I recommend you to please try to implement this, like write a pseudocode for this, and we can discuss this in the next class. All right. Any doubts about this one before I move on to new scoring schemes?
No, sir.
Is clear how we are moving? Basically, there are
So
instead of
just one
having,
question.
yeah,
So in the lower matrix, we just travel like between two nodes when we are moving. But
yes,
in the middle matrix, it's three different nodes that will be covered.
yes.
So why so?
Because in the middle matrix, you have to consider whether you are moving along this node or you have to actually see at each point you can either move in this direction, you can move into the lower matrix, or you can move into the higher matrix,
Yes,
right?
sir, that's clear.
At
So
huh. So
all
at
I'm trying
each
to ask
node you
is,
have three possibilities. You
yes,
can
sir.
go along this matrix, you can go into the new matrix, the lower or upper matrix. So there are three possibilities.
So then how is it like better than an n-cubed algorithm? Like how does it improve the time complexity here?
It improves the time complexity in the sense that you don't have to take all the possible routes. So when you have this n cubed kind of thing, you also would have to now say initialize, let's say you go here, right?
Yes, sir.
You go into this node and now you can go into this node as well, right?
Yes, sir.
These are kind of sort of forbidden transitions. So you will never actually end up calculating these values if you were to
Okay,
do the
so
simple
the
dynamic.
basic problem is when you have like repeated gaps or deletions. So
Exactly.
it mitigates that. Okay.
Exactly. Did you get that, Ashi, or no?
Yes, I did.
So basically, at each point of time, when you are moving in the middle matrix, there are three possibilities of movement. In the side and the lower and top matrices, there at each point there are two possibilities. Either you can have an incoming, right? Either you are going traveling down, or you are traveling out of the matrix, right? So you are not going from the lower matrix to the insertion, the other matrix, this one. So there are no transitions from the lower to the upper matrix. So those are forbidden, right? Because you cannot have, biologically speaking, one part cannot be having an insertion and simultaneously the other part cannot be having a deletion. Make
Sir, it's
sense?
clear. Yes, sir.
Yeah. So,
Yes,
and
sir.
the
Yes.
other, yeah, and the other thing that you must know is, given that this is how the matrices are getting uh, set up, the way that you will be starting to calculate or populate this dynamic programming matrix is also going to be different. So earlier you were moving, say, from top direction. So the first row and the top row was basically filled up, and then you were going down like this, right? So now think about how you will start to initialize these matrices.
So one more thing, like in this example, like the path is already given from like the previous examples, but in cases where the path is not given, so they'll again have to consider like several possibilities, right? So for example, if it
So the
starts
path
on the
can,
top left,
yeah, so yeah, but in this case, path is given because I want to show you how you would traverse this. But
yes, sir,
the path might not be given, right? So when the path is not given,
yes, sir,
then in that case, you have to start to initialize. So what can you initialize easily? You can initialize this one, right? You
yes,
can
sir.
initialize this one here. It's readily initializable. And then you will start to calculate the values of this movement. So the movement will still be like this. But just that when you start to calculate movements in this direction, at each point, you can either initialize a value in this matrix, or you can initialize a value in this other matrix. I suggest one thing. Let's say you are given the previous examples that we have discussed, right? Try to
Yes.
do this by yourself. Try to do this by yourself. Try to initialize this matrix. Try to initialize the values of these matrices and try to build the using the dynamic programming. Try to do this by yourself for a short sequence, right? Let's say I have a let's say this sequence that I discussed earlier, the two gaps, right? So this one is you score using the old one. It's quite straightforward, right? Make the dynamic programming like the dynamic initialize the matrix using this kind of alignment where there is no gap opening, and then do the gap opening one and do the alignment for this particular one. So I'm not telling you how it alignment would be done. I'm just telling you, so align the two sequences here, GAT CCAG and GACAG. Try to do this, and we can, if there is any
Nice.
issues, we can discuss this in next class. I mean, this would just take too much time for me to go step by step in each one of these. But the basic idea, I hope that is clear.
Sort of, sir.
We do it by hand, and it will become more clear. I mean, this is almost like a homework
Yes, sir.
problem. Yeah. So, okay, moving ahead, there are the two scoring matrices that I talked about, actually three kinds of scoring matrices. So one was based for protein scoring. So these are called PAM and BLOSSOM. And the other one is for nucleic acid scoring, and there are two models for that. one by Jules Cantor and the other one by Kimura. So the basic idea of these scoring matrices is that when you get two alignments, so for example, you have an alignment of two protein sequences here. Here you have two alignment of amino acids. So how would you score them when you align and sort of how would you score the alignment here? So whenever there's a P matching with a P, given a matrix like this. So for example, there's a T matching with a T given a matrix like this. It shows that whenever a T matches with a T, the score given should be 5, right? On the other hand, whenever a T is mismatching with a G, the penalty should be -2. And these penalties are not equivalent for between all the amino acids. And similarly, such kinds of penalties are not equivalent for all the nucleic acids either. So how is it that people have come up or are thinking about making these matrices is what these part of the lecture is about. So there are two kinds of matrices which are more involved for protein scoring. So I'm first going to talk about these two ones, which one is called a percentage accepted mutations matrix or PAM, and the other one is called Blossom, which is blocks of substitution matrices. So let's go for the first one, or percentage accepted mutations matrix. So this was suggested in 1978 by Margaret Dayhoff. So this was also called these are also called Dayhoff matrices. So what they did was you had lots of they had lots of whatever sequences that were available of common proteins. They had about 71 groups of proteins, which were at least 85% similar. So you have these 85% similarity. So if you take any two sequences, the number of mutations that you would have, or in like, and you align them with each other, they would have at least 85% or more similarities in the aligned sequences in pairwise alignments. All right. So now they took these sequences and they manually sort of aligned them and sort of got alignments like this using multiple sequence alignment tools, something that I'm going to talk about uh, after this. After they got this kinds of alignments, they were now having about if this was between 71 groups of proteins, they had 1572 amino acid substitutions or changes. So what do I mean by this? They were when you align these proteins, they were 1720 positions where you did not have a consensus amino acid, or there were mutations in the amino acid. So these are all representing uh, columns, right? So each column would be like an amino acid. So now after they did that, they essentially first,
Sir,
uh, yeah.
you said that the 1720, sir. So from where that number comes?
So this is the number of columns which will have mutations. So these are 71 proteins, right?
Yes, sir.
They would have certain length, right? So there are 71 protein groups, not 71 proteins. There are many more proteins than that. So they are forming in 71 groups, right? So each one of them group will be aligned like this. And then you are going to see, okay, how many positions you have amino acids which are different from each other. So for example, if I was to go here, you say that there are two positions where there are differences between nucleic acids here, right? After alignment.
Yes, sir.
So similarly, they will say after alignment, how many positions have a, uh, how many positions don't have actually a mutation. So the original letter is not the same. All right. So these are called amino acid substitutions. So the first thing that they do is they count the number of changes of each amino acid into each other amino acid in these locations. All right. So how do you do that? You assume you assume the consensus sequence. So you basically assume that what's the most common amino acid at each location, and from that amino acid, for that amino acid, you calculate okay how many times that amino acid was mutated into another amino acid in these 1572 amino acid locations. All right. Now after that, you calculate what's called the relative mutability. So you essentially say, okay, your amino acid was mutated n number of times, a particular amino acid across different locations, and this amino acid itself is present at a certain percentage in the sequence. So let us it's possible that some amino acid is not found very often, and some amino acid is found very often, right? In proteins. So the amino acid which is found very often in proteins that has higher probability just by chance of being seen, right, in such alignments. So you would basically uh, account for that, and you will calculate what's called relative mutability values. And once you have done that, you will compile all these values in a 20 by 20 matrix, and then you are essentially going to um, transform these values into this kind of penalty matrices. And I'm going to show you how. So the basic assumption for this uh, scoring scheme is that the changes that you're observed are between closely related proteins, because the proteins are having 85% similarity. So these amino acid changes do not significantly affect the protein function, because they are between similar groups of proteins also. So therefore, these matrices are called percentage accepted mutations matrix. They are essentially looking at mutations between similar proteins, or mutations which are accepted by nature. That was the idea. So how does it work? So I'm showing you an example. These are the this is one matrix, right? Where these probabilities have been calculated, the values have been counted, and the probability of having an alanine replaced by an alanine in the same sort of a column, right? Pairwise, it's calculated the probability and then has been multiplied by 10,000. So you get these kinds of probabilities. Now, the next thing that you do is you convert these matrices into what's called log odds matrices. Okay. So what do you do? You calculate the odds ratio for each substitution. So what is the probability of A getting substituted by, let us say, N? All right. So it will be 4 divided by number of times you see A, 4 divided by 9867, right? You basically divide the frequency of the amino acid, you take log 10 of that, and then you multiply that by 10. So it will be 4 divided by 9867. You take log 10 of that. So it will be approximately 1 divided by, so let's say this, take this one, R into A. This will be 1 divided by 10,000 approximately. So log 10 of 1 divided by 10,000 is how much?
-4.
It's -4. Right? And then you multiply it by 10 and you would essentially be getting the values like this. So now take the log once you would be getting, so you will have A converting into R. And so they say alanine converting into R here. And then you will have arginine converting into A, right? So you will have two of these log odds ratios, and you will be taking the average of these two log odds ratios. And then you will be basically converting into a matrix like this. So it will be, say, slight, not much difference here. So this value of A converting into R, let us say where is A? This is A, and where is R corresponding to A? See, R is here. So it's, this value is approximately -2. So it's not exactly conversion of this matrix into this matrix, but this is how these matrix values are converted. You first make these kinds of probability matrices, calculate log odds ratios, and then sort of adjust the values of those log odds ratios to get this kind of a matrix. Now, the assumption here is that the mutations that are happening are accepted, and these mutations are independent of each other. All right. So since these are independent mutations, so each event is independent of each other. So you can actually now, based on a simple matrix, the first matrix that they created is called the PAM1 matrix. So PAM1 matrix essentially says that we found one mutation for every 100 amino acids that we looked at. Okay, so we looked at uh, those proteins, those protein groups or those sort of locations which were giving us one mutation for every 100 amino acids. So remember, we have lots of such alignments, right? Lots of these cases available, and you can sub-select which cases you want to work with, which alignments you want to work with. So if you work with alignments that are on the order of giving you one mutation for every 100 amino acids, so that essentially means that you are looking for very few changes, right? So for every 100 amino acids, only one amino acid is changing. So these sequences which are underlying these alignments are very similar to each other. So you can make what's called a PAM1 matrix. right? Similarly, you can make a PAM10 matrix where you can say there were 10 mutations for 100 amino acids. That means the alignments were having more mismatches amongst themselves. So, and so on and so forth. So you can go to PAM250, which says there are 250 mutations happening for every 100 amino acids that were observed, 100 alignments that were observed. All right. So like this, you can.
Sir, I have a question. So, sir, this PAM1, PAM10, PAM 250, these matrices are constructed based on the sequences we have, or we can use these matrices for any kind of amino acid sequences.
So that's what I'm trying to
Uh,
tell you. So this is what these matrices mean, right? That you are having one mutation for every 100 amino acid. But the thing is that typically you start with something like with a PAM1 matrix, right? Where you look at all the amino acids. Now, because PAM1 matrix, the assumption is that each time you have a mutation event, it is independent of the other mutation events. So the PAM1 matrix can be multiplied n times to give a transition matrix. So if you multiply PAM1 with itself 10 times, then you get a PAM10 matrix. So you don't actually have to look at specific columns because you only construct the PAM1 matrix, right? And you get this kind of scoring scheme, which is converted into intermediate scoring scheme. Let's say that's why it was -4, right? And this one, what the matrix that I'm showing you here actually is the PAM 250 matrix. So you've multiplied this matrix that was giving you the log odds ratio and by itself multiple times, 250 times actually, and you ended up with a matrix like this. So you basically the assumption here is that the PAM1, because the matrix, the changes are independent of the previous mutational events, you keep on multiplying itself. So the mutations are going to be the scores are going to be uh, going more and more different from each other with each multiplication, and therefore you will be representing more distinct sequences. So this is like sort of created. So essentially, when you are looking at more similar sequences, you would go for a smaller PAM value. If you're looking at more distant sequences, sort of comparing more distant sequences, then you will go for more larger PAM values. But obviously, it's difficult to explain to you the idea behind it. It is difficult to also understand, right? So therefore, people started to come up in much later, 15 years later, they came up with the idea of Blossom, which is block substitution matrices. So this was put forth by Henikoff, and this had a much simpler idea, an idea that is makes much more sense and does not depend on this kind of derivation of mutational rates. So what they did was they derived a database of local alignments of distantly related protein sequences. So they took protein sequences which were distantly related, and by that time you had databases like ProCite, which were giving you protein families. So they took a large set of about 2000 amino acid sequences that were organized into blocks. So what do I mean by blocks? You have these protein sequences, right? You align them locally, and we know that proteins are composed of conserved domains. So the conserved domains between proteins will be aligned. All right. And these blocks and each alignment of such a block is basically each alignment of such a domain kind of a structure is called a block. All right. So for example, here was domains, you align them and you got a block. Now, after you've got a block, you calculate how often do you see a pair of substitutions. What do I mean by that? So let's say we have a block here with 5 sequences. There are 2 locations. There is a A, 2 locations. There is a C, and 1 location. There is a E. All right. So what do I mean? So basically, if I took any 2 random sequences, if they initially had a A, right, then there can be 4 possibilities where the A was getting mutated to a C, because the first this sequence could go to a C, this sequence could go to a this C, right? Or this A could go to this C, or this A could go to this C, right? So there are 4 possibilities or 4 pairs of transitions from A to C. Similarly, there is two transitions from two A's to a E, two transitions from a C to E because there are two C's, and there was one transition from A to A and one transition from C to C. Is this clear?
So could you explain the transition part again?
So the transition
Uh,
part is, so let's say you have these five sequences, right? These are five
yes, sir.
underlying sequences in this particular block. So we are basically trying to calculate what is the frequency with which A becomes a C, right?
So,
You
but
can
why would it be 4 here?
it will be a 4 because this A can get mutated into the sequence context of this C, because now we are talking about mutation of one sequence, this sequence here. So this top block into this bottom block, right?
Fine, sir. Got it. Got it.
So
Got it.
there'll
So
be a
it's
mutation
basically 2
event.
into 2, right?
There'll
So
be 2 into
first
2,
egg
right?
gets, yeah, okay, got it.
Exactly, exactly. So the assumption here is, evolutionarily speaking, is now whenever we are talking about these kinds of mutations, we are talking about how does one sequence convert into another sequence, right? So if we are talking about this first sequence, which is an A, this can be converted into this kind of a sequence, third one, or it can be converted into the fifth kind of a sequence, because here we are not only concerned about just the amino acid here, but we're also concerned about the context of the amino acid. Where is the amino acid lying, right? So similar, so this A can be converted into these two different kinds of C's. This C, which is represented in this kind of a sequence, or this C, which is represented in this kind of a sequence. Similarly, you have to do this for each A. Is this clear to everybody, this part?
Yes, sir.
How we are getting 4 here?
Yes, sir.
Yes, sir.
Okay. So now what do we do? In order to calculate this kind of a score, you basically you calculate the log odds ratio, which is basically the probability of observed pairwise, the observed appearance of the pair of the two amino acid pairs. So for example, C and A, it's 4, and the expected appearance of A and C. So the expected appearance of A mutation to C, what would that be in this case?
So 4, right?
Sir, can you repeat the question?
So there are, out of 5 sequences, A is appearing 2 times, and out of 5 sequences, C is appearing 2 times. So if this was happening randomly, so what would this expectation be? It would basically be the frequency of AI multiplied by frequency of AJ, right?
Yes, sir.
So basically frequency of this was 2 by 5 multiplied by 2 by 5. So 0.4 into 0.4, right? That's the expectation because it is happening randomly, but
Yes,
actually
sir.
this does not happen randomly. So you see that A to C is 4 out of the 10 observed cases. So it's actually zero point 0.4. And if you multiply zero point 0.4 multiplied by zero point 0.4, this value is 0.16.
0.16.
So the log would be a positive value, log value here. All
Yes,
right.
sir. Just one a small question, sir. I could not understood like why A is above C is C. The two A are above C. Why we have kept that above?
I'm just taking it from here, this block. So these are protein sequences which have a common domain. All right. We align them. After alignment, we look at what are the blocks, what were the amino acids that were which were coming here, right? So in this particular alignment, the amino acids, this AAA, so these are amino acids at that particular location in the block.
Okay, sir. Understood. Understood.
I'm just taking the same thing here for the next slide just to show you how you would convert this into a blossom kind of a matrix. Okay, so now earlier in the case of PAM, we calculated PAM1 and we multiplied it by a multiple itself. to create PAM10 and PAM250. But in the case of Blossom, we actually have the possibility of looking at blocks, looking at different blocks with different kinds of uh, substitution rates, right? Different kinds of similarity. So basically, there would be some blocks or some domains which will be highly similar between proteins, right? So, and there will be some domains which will be not so similar. So now in order to distinguish between these two cases, what people did, they suggested that you would basically create Blossom N matrices. So Blossom N essentially means whenever you have a block with N% identity to the consensus, all right, then you would call it Blossom N matrix. All right. So for example, a Blossom 62 matrix is going to come from an alignment or a particular alignment of a block where 62% of any of the, so basically the consensus sequence is going to be representing. So here, can you tell me what would this be? If we just look at this, but these 5 amino acids, what kind of, and we are only talking about just this particular location, what kind of Blossom matrix would this be representing? How much percentage identity does it have to the consensus sequence? So what is the consensus at this location?
A or C?
A or C?
A or C.
Yes. And how many sequences are equivalent to the consensus?
20%?
Right?
40%. Yeah.
40%. Yeah.
40%. So this basically this alignment represents a Blossom 40 alignment. And so if you would have, so you will calculate basically an average identity between all the columns, and corresponding to that average identity, you will basically come up with a appropriate Blossom matrix. All right. So the choice of amino acids, the choice of the proteins chosen here, and the block or the domain chosen is going to give you the kind of matrix that you want. So if you want to work with proteins that are more distant, so proteins which are more distant are going to have less identity, right? With the consensus. Therefore, they are essentially going to represent mutational rates between more distant uh, more distant proteins, right?
Sir,
So
I have a question.
a smaller number means there is a larger evolutionary distance between the proteins. A larger number means that the underlying proteins or the underlying blocks used to make this Blossom matrix is more similar to each other, is having smaller evolutionary distance. Yes, Ratnesh.
Sir, okay. So, sir, what's the difference between identity and similarity?
So identity is with respect to uh, a consensus sequence. So identity is in multiple sequences, you try to see identity, right? So we are basically saying 62% identical amongst lots of sequences. Similarity is between two sequences. Similarity is two sequences are similar or not.
Yes, sir. So,
Similarity is
like,
between at the level of two sequences.
yes, sir. Like, I have two sequences and I put them in what is these tools which is available for local alignment and global alignment at emboss. So they gives identity and this much percentage and similarity this much percentage. So from where they are getting this identity percentage?
I can take a look at this and I can get back to you. So identity percentage is typically done from the consensus. So identity is the averaged out value and similarity is base by base value. Identity is typically looked at large in the context of larger sequences. I will confirm exactly what Emboss is reporting and I'll get back to you on this.
Yes, sir. Yes, sir.
Whenever you're basically talking about scoring matrices, there are two: PAM, right? So PAM is basically matrices uh, which are made from this made by this older algorithm where you take the protein global alignments of protein sequences of closely related protein sequences, and then you essentially say that these alignments are representing tolerated mutations. And so you start with, say, PAM1, which essentially represents protein sequences that are similar between organisms that are 1 million, represents 1 million years of evolution. And now you multiply it by itself to extrapolate to larger evolutionary distances. Blossom is derived from local uncapped alignments of distantly related sequences. You can basically uh, choose the sequences that you work with. All the matrices that you have are directly calculated. There are no extrapolations here. And the number of after the matrix represents the minimum percentage identity of the blocks, right? So the blocks that I showed you used to construct the matrix. So greater number means there are lesser distances. That means there is more identity. So Blossom series of matrices typically is the newer ones perform better because they are not extrapolated like in the case of PAM. So this is how it looks like. A PAM1 is similar to Blossom 80. So PAM1 is basically corresponding to proteins which are very similar to each other. PAM 250 is corresponding to Blossom 5045. So basically representing 250 million years of evolution. And typically you will see in a lot of your tools that you will be using in the next module, for example, for multiple sequence alignment, you will be using tools that will be taking Blossom 62 as the matrix, which is corresponding to PAM 120. So which is basically capturing 120 million years of evolutionary divergence. All right. So when you are comparing, a general tip is when you are comparing closely related proteins and you want to find closely related sequences, you use a lower PAM or a higher Blossom matrix for your scoring penalties. And if you want to look at distantly related sequences, you use a higher PAM or higher or a lower Blossom, which would tolerate more mutations in the alignment. Is this clear?
Yes, sir.
So the
So
scoring
one more thing.
matrix, yeah,
So these models are just for comparing like the evolutionary similarity, right? Not the functional similarity.
not the functional similarity. So there are other matrices which are based on three-dimensional structural conservation and functional similarity, which people are now working at.
Fine, sir.
Yes, sir,
Pranay.
sir, why can't these be used for functional similarity in case we are using this? All these scoring matrices are being constructed based on this mutation rates, right? So
Yes.
that's why.
So the functional similarity here, we are assuming the basic assumption for making these matrices is that amino acids presence, right? Similar amino acids, similar sequence means similar function. That is the assumption here. But it is possible and it is known that two proteins which might not have very similar amino acid sequences might still perform similar functions. So therefore, if you want to, what's the idea here? If two proteins are having similar functions, ultimately, you want to find a third protein which looks like these proteins, right? Whenever you're doing these kind of alignments, right? You have a new organism in which you want to find, let us say, a DNA polymerase, right? Which is doing DNA replication. You have information about all DNA polymerases which are out there, right? You want to use this information to now find a new DNA polymerase. All right.
Yes, sir.
So now in this case, should you, if you just take the matrices constructed in this way, your assumption is that sequence conservation is going to lead to similar function, which is the case, which is the basic assumption in biology. But there is still some cases, some outlier cases, some rare cases where sequence can be different. So this is basically something which is coming up. So this is an active area of research to make matrices or scoring schemes which are based on function rather than purely sequence.
Yes, sir. Like in case of like we saw that in tyrosine B1, so that can't be done like in local alignment, etc.
Yeah. So to end the scoring matrices, so there are two models for scoring nucleotides. So nucleotides are rather similar. So one model is called the Joule-Cantor model, which basically says that if you go from one nucleotide to any other nucleotide, this substitution rate is common. So this is basically alpha, which is a common base substitution rate. And the other model is called the Kimura model, which says that when you are having, there are two kinds of substitutions. There are substitutions called transitions, where the ring size does not change. So it's basically a transition from pyrimidine to pyrimidine or purine to purine as represented by alpha, right? So A alpha here, or it can be from purine to pyrimidine. So this is basically change in the ring size uh, of the nucleotide. So basically you have a nitrogen ring, right? So purines have double ring and pyrimidines have a single ring. So the single ring changes should be penalized differently from no ring changes. So what is seen in nature typically is that the transitions, the rate of transitions, right? The pyrimidine to pyrimidine and purine to purine, this rate is observed more often than transversions, which is basically change in the ring size. So therefore, they come up with two models whereby they essentially penalize transversions more heavily than transitions because they are less observed. So you should penalize them more. All right, this is the idea of the scoring matrices. So I'll end up with the drawbacks of dynamic programming so that we can move on from this to multiple sequence alignment. Dynamic programming approaches give you definitely guarantee you an optimal alignment between two sequences because this is a mathematically derived formulation. The drawbacks is that it is typically compute intensive and memory intensive because you have to maintain these large matrices and you have to have these backtracking pointers maintained, right? So it at least takes on squared in terms of space, right? And between on squared and on cubed for compute time. There are some linear space algorithms which have been used to deal with the drawback of dynamic programming. But the basic idea of these are to concentrate on only those areas which are more likely to give you maximal alignment. So how do you find the areas which are giving you maximum alignment is something similar to what you do with dot matrices. So wherever you try to have these kinds of diagonals, small diagonals which are off the main diagonal, those are the areas more likely to give you maximal alignment. And these are the ones which are typically used to save time in the case of like linear sort of more linear alignment. So you just start the alignment in those locations. And yeah, one of these examples is called the Myers-Miller algorithm. But the problem is that it is compute intensive because you have to first find the areas where you're expected to get a maximal good alignment. And this is the basic idea of that will be even used by alignment, which is most often used by most of you, must have been used by many of you, is called BLAST, which essentially tries to first find locations where you are more likely to find a local alignment based on what's called heuristics. And I'm going to come to that in the module in 2 weeks time. So I'll stop here. And Ratnesh, I'll come back to you with a question of how does the similarity and identity is calculated in the emboss package when it gives you results. I can take questions or otherwise you're free to go for the next class. Thank you for attending.